So the next topic is machine learning on graphs, and Arjun's going to coordinate that.
Thank you, Taylor, and it's been great so far, and to listen to all these topics. And many of these topics I've had not worked on for many years, and it's good to listen to all these things and refresh my memory. In our group, we're going to be talking about machine learning on networks, and then hopefully I'm obviously going to say many things that are going to be related to many other things that we've already heard during the day. And in addition to the people here, Sardar, Yang, Byunjan, hi on you, I'm also probably, I should have added Marinka's name here because I'm going to cite much of our papers also. So with that, I want to, here is what we're going to do. This is a lot, so we're going to see how far we can go, and then based on that, hopefully, cut some of the last parts. I'm going to introduce a very general framework to think about machine learning on networks. Not going to go into pros and cons, what is better and worse and all that stuff. I'm not going to talk about that stuff. And then we're going to talk about unsupervised and self-supervised training, condition-specific embeddings, and doing embeddings on heterogeneous graphs. And then Byunjan and I are going to talk a little bit about transfer learning, and then Haiwan, and then Yang are going to talk about geometric networks. And then we're going to stop at that point, take some questions, and see how we are doing with time. Okay. All right. So the idea of, so just to introduce this idea of machine learning on networks, I want to begin with this very simple overview that all of us, I think at some point or the other, are interested in the fact that there is some graph of nodes and edges, and then we want to do some task, classification task, right? Maybe classify nodes into discrete labels, or give a continuous number to it, or classify subgraphs or whole graphs, and then or do community detection, link prediction, or do graph alignment. These are problems that all of us heard during this day. And the traditional idea is to take this graph and then do feature engineering by hand. We basically figure out what is interesting about this network, or for example, each node, maybe its degree or its graph-like spectrum and all that stuff, and use that as input to machine learning algorithms and potentially make these, do these tasks. What instead might be a good idea is to actually take this graph and then learn something like a numerical representation for every single node in the graph. What numerical representation essentially means is a vector of numbers. That single vector represents every single node, and that vector uniquely captures the structural or topological location of this node with respect to all the other nodes in the graph. Okay. And if we are able to learn this structure, then we can use traditional machine learning algorithms to do any of these problems, potentially. The way to think about this in a very general way, what we can do is to design something like an encoder-decoder system where we have an encoding function. This can be whatever it is, in a way, I'm just talking about this in a very general way, that learns this numerical representation. It's one vector for every single node in this graph. Okay. And then we train a model that takes this graph as input, learns this intermediary embedding vector, and so that you can do this decoding to recapitulate a node's position in the underlying graph, or maybe some other label that we have on top of every single node. If we do that, once we are done with this part, we can take this embedding vector, and that tells us some kind of a numerical encoding for every single node in the graph that we can then use as input to ML algorithms. Okay, so generally, what all these algorithms are doing is to minimize some loss like this. What they're essentially doing is that if you take the embedding vectors of two nodes I and j, that's zi and zj, and then run it through this decoder, and then we have some sense of how close those embedding vectors are. And if how close two nodes are in the embedding space and how close they are based on the underlying graph, if that difference is as low as possible, then we have learned a good representation of all the nodes. How this distance of the original graph is defined, how this decoder is defined, and what this loss function is the way in which all these different methods vary. For instance, there is this class of methods called matrix factorization methods. And what they essentially do is to say, once you have an embedding vector for a pair of nodes, you can combine them in two ways. Maybe do a dot product or whatever it is, or calculate a Euclidean norm. And then that will be the way in which you're going to decode the embeddings in terms of a distance between two nodes. And then we're going to define the distance between the two nodes based on the original graph in many ways. For example, you can just say two nodes are connected to each other based on the adjacency matrix. Those two are close and otherwise not. And then you can define a loss function that says how far the distance based on the embeddings is from based on the original graph. And then minimizing that will essentially give you give you a way to optimize the embeddings. So that's the matrix factorization ones. The ones based on random walks essentially say that we're going to define the distance between any two embedding vectors in the following way. We're going to say that we're going to do random walks on the original graph. Either they are straight up random walks or they are like biased random walks. And then once you do that stuff, what we are essentially asking is the distance between any two nodes is that if I start a random walk on node I, how frequently will I reach node j? Okay. And if that number is high, we want the corresponding embeddings to also be very close to each other. And that's what methods like deep walk and node to vec optimize for. Okay. So some problems is that methods like this do not actually share parameters across all the nodes at the encoder phase. And second is that this does not take into account any additional information we might have about nodes. For example, let's say the expression or activity level of all the nodes in the graph and stuff like that. And also, they are intrinsically constructive. It means that once we have learned node embeddings for a given graph, we can't predict the embeddings for a node that has never been seen during training. Okay. So there are approaches to sort of address all these problems, and that is by way of using graph neural network models. And the idea of graph neural networks comes from the idea of convolutional neural networks on images. The fact that you don't want to treat, for example, all the pixels in the image as totally independent of each other. There is some spatial relationship between all of them. So you can think of this as a mesh graph of all the pixels. The same way we have nodes, and then they are all arranged with each other based on the underlying graph. And there's been a lot of development where people have sort of adapted this idea and then turned this into message passing. And then some of the latest ideas is based on graph attention. Okay, so the idea here is that one simple way of thinking about deep neural network models on underlying graphs and to learn node embeddings is this idea that you can basically train an autoencoder model, for example, that takes the adjacency vector of any given node and learns a low dimensional representation so that it can be reconstructed back. Okay, and then that low dimensional representation is a node embedding, and that's not a bad idea. However, this doesn't, again, take into account, it's still constructive, and you cannot do this for any new nodes and all that stuff, and it doesn't take node attributes into account. So the more modern idea is to say that you have an underlying graph, and then you do this combination aggregation based on the network structure in a recursive and sequential way so that you can keep updating the embedding vector of A over time, and what you learn over time is the best representation. Okay. Let me just say one small thing about this in the interest of time. Again, all the graph neural network based algorithms all fall roughly into this very general framework. And what they essentially differ on is how this aggregation is actually done. Those aggregation functions are different for different neural network models. And then how these weights are actually shared across all these nodes. These weights are actually learned during every single time. And this aggregation combination. So these are the ones that are actually different across these different networks, different algorithms. And okay. Maybe one other thing that I want to say is that in the simple case of graph neural networks, what we have is that let's say we take a single node, at the aggregation step, what happens is that you sort of treat the value of, or rather the embedding vector of all its neighbors sort of equally. But that itself is an assumption. So you can sort of say, okay, maybe you want to build another neural network model that says how to weight the contribution of all the nodes of all the neighbors of a given node, and then how that's actually aggregated to update the embedding for a given node. That is called attention, and that's been proposed for graphs. And then finally, this is my last slide for this introduction, which is that in addition to up to this point, what I've essentially said that you have a graph, and then you can ultimately learn an embedding. But what if you not only want to gently learn an embedding, but you want to learn this embedding in the context of a supervised classification task you are interested in? Let's say you want to do, disease gene prediction or function prediction. In that case, what we would also want to do is to add another loss function that basically says how well you're doing with this specific classification task. Add that to your embedding loss as well. And then you can learn this in a supervised way. Right. Okay. And then, Yang, you want to go next?
Thank you, Arjun. Okay. Okay, thank you. So, so as Arjun have introduced the concepts, and the first topic we want to talk about, which covers about 20 minutes, is embedding. This topic has already appeared in this morning. I feel like it has a lot of interest in our group. So let's spend a little bit of time on this part. So I will talk about the first part of embedding, which is unsupervised or self-supervised pre-training. I'll explain what I mean here. So far, Arjun has shown that you can use matrix factorization or random walk, right, to learn the network embedding. Then recently, we can use deep learning, graph neural networks with the help of the labels. We might be tempted to use supervised learning for graph embedding because we feel like it's learning not only context-dependent, but also task-specific embeddings. But the problem of such a supervised approach is apparently the curse of the labels, right? Especially in our fields, in biomedical or biological discoveries, the labels are very expensive, right, and rare. So if we recall what happened in computer vision in the early days, they can use crowdsourcing to label images, right? Here, it's really hard for anyone, right, to mark the tumors on the brain image. But you see, very often from reCAPTCHA that you are asked to label a traffic lights, right? So it needs a lot of domain knowledge in our field to generate such label data. Not only the amount of the label data is rare, but also in our field, the label data can be very context and procedure dependent. So speaking of how two proteins bind to each other, you could have different kind of assays to measure them, and then each assay have different conditions and so on. So it's very subtle. So, but the challenge is more than just that, the labeled. The additional challenge is from how we want to use embedding, what kind of embedding we want and how we want to use them. So if we do even manage to use a small amount of labeled data to learn the embedding, there are two issues that are central to our use of the embedding. First of all, it's really hard to generalize. So if you want to explain what we have learned from the embedding through visualization or other methods, or you want to transfer the embedding for a different task, it's very hard to use those embeddings to generalize. And the second thing is the robustness. So we have a lot of noises in our data, not just the labels, but also in the graphs themselves. So under such uncertainty, it's a very poor embedding that we can use from the supervised approach. So if the supervised learning is not the answer, just like people have already called that the next revolution in AI is not supervised, then where's the answer? Well, we can all guess if it's not supervised, then it's unsupervised, right? So I'm going to talk about self-supervised learning, which is, just like unsupervised learning. It's overcoming the bottleneck of labeled data by not using labeled data. Essentially, we are using unlabeled data, and what's even better is we have massive amount of unlabeled data. We don't have a lot of protein sequences with structures determined, but we have so many protein sequences, right? So in this case, what is self-supervised learning? It's a branch of unsupervised learning. It's trying to use part of the input itself to predict the rest. And in this example, you can see that this is an image example that we can design some so-called pretext tasks, which only involves the input itself. And then the encoder can learn some parameters, and that can be the specific form of the knowledge transfer to the downstream task. So this is a concept of the pretext versus the downstream task. So specifically, let's look at what is the pretext tasks. I want to show you from the left to right are three domains circled. One is a computer vision, CV, one is a natural language processing, NLP, and one is graph. So actually this pretext designs or self-supervised learning started with computer vision and NLP. So you have top and bottom rows representing two styles of self-supervised tasks. On the top, you have the so-called predictive kind. And as you can see in one example, right, we start with the image, we make it black and white, and then try to predict its color, right? And in the bottom, we have the contrastive kind of methods. So we start with images of different fruits, but then for each image, we generate a new view, or we augment the image by rotating or do some other kind of perturbations. And then we construct positive pairs and negative pairs. We assume that the different banana images are positive pairs, but across banana and apple is a negative pair. And we try to learn embedding to make positive pairs close in the latent space, but the negative pairs far away in the latent space. So the same idea has been propelling the natural language processing through the so-called masked language prediction, right? Masked language modeling. So these trends have recently rippled to the graph field, and I'm gonna show you two types of methods in more details, but the fashion is very similar. So for the first kind of predictive models, right? So these are two kind of examples, right? One is graph reconstruction, just like the name suggests. You try to hide part of the graph and predict the rest of it. Here you can hide part of the nodes or edges, and use the same encoder-decoder architecture that Arjun has shown. And then this is one type of methods. You can also collect something that's in the input data, but not exactly part of the input data. For instance, you could summarize the topological clusters or communities in the image and then try to ask the encoder-decoder system to predict where these nodes belong to different clusters and so on. So you could use this input data itself to design different kind of pretext tasks for the self-supervised learning. So this is the first type, predictive. In the second type, self-contrastive methods, I'm just pointing out two important factors of contrastive methods. First is how do you generate the augmentation or views of the original graph. So apparently in the middle panel, you see three kinds of perturbations, right, involving the node attributes or topology or sample perturbation and so on. And the second factor is how do you contrast the positive versus negative pairs? And you could use different objective functions to pull apart the two distributions. And in the end, I want to say a little bit about the challenges and involving this self-supervised learning. So first of all, extending from images and languages to graphs is a very non-trivial task for self-supervised learning because the data structure is different. Unlike images, which are grid data, here we are facing non-Euclidean data, and we have all kinds of things, we all call them graphs, but they are abstractions from diverse natures. So it's very non-Euclidean and heterogeneous. And in the open challenge is that, first of all, for the predictive methods, how do you design the predictive tasks? There's not a lot of universal framework about it, and people are using different kind of methods. And then what about the contrastive methods? What kind of view do you generate, right? So we try to optimize view among a pool, or we try to even optimally choosing a view in a continuous space by graph generation, but it still is an open challenge and a lot of things ongoing. And lastly, how do we better incorporate our domain knowledge into the design of the self-supervised tasks? And that's very important. So essentially, these self-supervised tasks are ways we incorporate or impose our human information into the prior of the graph. Of course, there are a lot of other topics as well, including explainability, which will be briefly touched upon as a separate topics. And then you might have other more complex graphs that my two colleagues will talk more about, Arjun and Serda. Thank you.
All right, I'm going to briefly touch upon our next topic, which is on building condition-specific network embeddings. Okay. And the idea is that we not only want to say that all of us, we discuss a lot about dynamic graphs and the fact that graphs are not static. There are potentially, the graphs are varying a lot in terms of various conditions, both in terms of node membership and edge membership. So the idea is to say, now, why don't we assume that we don't only have a single graph, but we have some notion of a context, and then can we incorporate that into network embedding? And I'm just going to at a very high level talk about some ideas. So one is that, for example, the best way, one good way to think about context-specific network embeddings is to just begin with context-specific networks. So for example, if you are able to generate, for example, we generated a long time back gene interaction networks that are specific to different tissues in the cell type, in the human body. And then Marinka basically took this, did a really nice work taking these networks and then incorporating that into a single model where every single layer here, for example, corresponds to a specific tissue network, and the nodes are individual genes. And then there are connections between the layers based on how similar these tissues are, based on anatomy and physiology. And the goal is to optimize learn embeddings so that you optimize two things. One is that you want nodes within a given layer that are structurally similar to have very similar embeddings. And you want the nodes across, the same nodes across layers to have very similar embeddings if those layers are also pretty similar to each other at a high level. And you want to optimize both at the same time, and then they did this nice work, and then you were able to get one embedding per node per network. And that's been shown in that paper to be useful for function prediction and stuff like that. What if you don't have one network per context to begin with? So what we are exploring now, for example, we are sort of wrapping up this work where we assume that we have a single network. But then for us, context information comes from the fact that we have some score for every single node that has been produced experimentally. For example, you can think of this as a bulk or a single cell RNA-seq profile. And then the way we handle this to say, we're going to take a single profile and use that to modify the edge weights, and then use that to influence the random walks that essentially build these node embeddings. So therefore, we end up with context-specific node embeddings, and we initially see that these context-specific embeddings are much better than the original embeddings that are devoid of this context in making meaningful predictions. I also wanted to highlight another figure from Marinka's review, and this is not exactly context-specific network embeddings, but I'll talk about how it is presented here. So which is that, Marinka, correct me if I'm wrong, okay? So the idea here is that there is an underlying graph which is static, and then let's say you have gene expression data. Let's say for every single cell type, it's not like gene expression, but let's say a set of genes that are enriched in that particular cell type. So what you can do is to do the supervised node embedding, which is to say you take this entire graph and then learn a network embedding that maximizes the ability of the embeddings to predict whether a gene is expressed or enriched in a particular cell type or not. Okay. So that is the use case that is presented in this review, but I thought this could also be used to actually generate context-specific network embeddings, where you can even think of this vector of which cell a particular gene is present in as node attributes, and then use that to potentially even generate context-specific embeddings. So that's something to think about. Right. Okay. Sarah?
All right. So in the morning, when we discuss about multimodal data integration and knowledge bases, we talk about different data modalities, and multiplex heterogeneous networks are good ways to represent different data modalities. So you build one graph where you have multiple types of nodes, multiple types of edges, and now the problem becomes, okay, can you do graph representation learning on this complex graph? And there are different application domains. This could be a recommendation system where you link prediction between customers and items, or drugs and the genes, and different things. So There are different names. Multiplex heterogeneous network is one name. Multi-view networks, heterogeneous information networks are some of the names that you can see in the literature. But essentially, its total number of node types and edge types is greater than 2, then we're in this territory. Again, when we do machine learning, we can categorize into two main groups: node classification and link prediction, and drug reproposing, drug side effect prediction, gene prioritization could be some of the examples of node classification. Drug target prediction, recommendation systems, fraud detections are some of the applications that are in the literature for link prediction using multiplex heterogeneous networks. So as in the regular graph representation learning, there are two main streams, shallow learning and deep learning. And the disadvantages are really similar to what Arjun explained. And so I'm not going to go too much into detail. For the shallow learning, I'll give some examples. So there are two main steps. One is how we're going to define the paths for the nodes that we are trying to embed. And as we discussed several times, we do random walk based methods. And basically, when we do random walk, we could do, starting from a seed gene, we can visit other nodes in the same layer, or we can jump to the other layer. So it's a random walk with restart on multiplex heterogeneous networks. We can also define method paths, where we're doing, again, random walk, but we are particularly careful about what kind of nodes we visit. So on the graph on the left, so we can define a similarity between two author nodes based on their co-authorship. So we can visit paths starting from author, go to the paper node, and then author node again. Or we can look at the author similarity in the context of presenting at the same venue. So then our method path would be starting from an author, going to a paper, going to the conference or journal that paper was published, going back to another paper, and then another author. So this kind of paths can be defined. And then the next step, we can apply like the skip-gram algorithm that was first introduced in word2vec, and then node2vec was applied. There is heterogeneous skip-gram algorithm where the idea is at the output layer you categorize them based on the node type, or you can define a similarity probability distribution on the graph and also the embedding space, and you can compute KL divergence between these two probability distributions to optimize your embedding. But the limitations are, again, as we discussed earlier, there are node attributes that we could utilize, and shallow methods cannot do that. Method path definition requires some semantic understanding of the graph, and if it's a more abstract graph, then it's hard to do. Number of nodes and edges could be limited depending on what method you use, or the edge weights could not be utilized. But more importantly, as Arjun mentioned, these are transductive methods. So every time you have to start from scratch to learn an embedding. And there are some hyperparameters, like what will be your jump probability or restart probability. It's hard to tune those hyperparameters. So the deep learning methods can overcome some of those issues. I'll just talk about some kind of historical perspective on some of the methods. So heterogeneous graph neural network is one of the first approaches, applying GNN on heterogeneous networks. So the node attributes, there's an aggregation for the node attributes, and node attributes in this method are coming from different data modalities. Like it could be image, text, so on and so forth, so they can be combined. And also you do aggregation at the node type level. So you learn embedding at each node type, and then you combine them together. So again, Arjun mentioned about graph attention networks, and the same of that are applied to heterogeneous networks. So heterogeneous attention network is another method. Again, we do aggregation. Aggregation is done at the method path level and at the node type level, but then you compute attention between the nodes when you aggregate them, and then you also do attention between the method paths in this method. Another popular method is GATNE, which has two versions. So there's transductive version, but there's also inductive version where you learn a transformation function so that you can apply that function on a new graph that's never been seen. Again, there is attention applied, and the optimization of the embedding is done by using skip-gram algorithm, like in method path to vec method. And the final example that I'm going to give is just published recently. This is a self-supervised method. So there's contrastive learning, as Yang mentioned. Contrastive learning applied on heterogeneous multiplex graph. Again, we have the multipaths introduced here. We do graph sampling based on those method paths. So we could call them method graphs. And we do aggregation, again, for each of the method path. Separate aggregation is done. So they call this intra-learning, intra-contrastive learning. And also between the different semantics, you do aggregation. So it is inter-semantic contrastive learning. So they are all combined, and you have a final loss function to learn these embeddings. Okay, so some of the challenges, some of the open problems are, again, here we also mostly rely on method paths or method graphs, but understanding which paths are relevant really depends on the semantics. So we might want to focus on network motifs as well, to be able to apply this on more abstract or graphs where the semantic information is not readily available. There are also dynamic networks, as we discussed in the previous session. So networks that change over time. So how we could apply these methods for dynamic networks. And like GNN, we know that GNN suffer from over-smoothing problem. Is this same thing happening for the heterogeneous networks? That needs to be studied a little further. And something that we also, we're going to talk about explainability a little bit, but there's also how to build those models in a fair fashion, meaning that you utilize some node attributes, but those node attributes, like there will be some sensitive node attributes as well, like demographic values. And if you don't have enough sample data, you might build a biased network, maybe biased to different race or different sexual orientation. So that learning them in a fair manner is also an important future direction. These are the papers that I cited in the slides. So I think Arjun or Arjun? Oh, okay, you're here. Well, great.
Yep, finally. I was the last person. Very nice meeting you. I will talk about transfer learning just a little bit. I think transfer learning techniques could be a very useful class of machine learning techniques that could, make big contributions to, network biology in the future. I'll spend the next 2 slides on, oops. It's not this one. Oh, this one. Okay. Oh, great. Yeah, so many of you may be already familiar with transfer learning, but let me just briefly summarize. Transfer learning aims to take advantage of the learning outcomes for a specific predictive task in a specific system, and then try to apply the learning outcomes to a different domain or different system or different predictive task for the same system. Okay. Why is it important? Because, well, everyone that works in computational network biology knows that, well, we already, don't have enough data, right? And considering the complexity of the biological networks that we have to deal with and the predictive tasks that we want to do, well, how do we solve this problem of having insufficient data, especially for non-model organisms, for example, right? One thing that we could aim to do is, well, make the best use of existing data and see how much it can be transferred for different predictive tasks or different systems. And that's what transfer learning is trying to do. And in a sense, at a high level, we've been already trying to do that as computational biologists, right? Well, make the best use of what we already know, and then try to apply it to different systems of tasks. And machine learning could be, well, used to do this in a more rigorous and formal manner. Oops. So, you may have heard about domain adaptation. It can be viewed as a specific type of transfer learning, where we make some assumptions about, the feature space. For example, there may be a space of features, and then you may use those features to predict something or do some kind of analysis. And when you think about the same feature space, however, with a different distribution of the features, you typically say that you want to adapt the domain, right? So it's called domain adaptation technique. Transfer learning is conceptually a bit more general, so you don't necessarily have to assume that the feature space is identical, okay? So, how can transfer learning be applied? For example, it may be used to answer the following question. For example, suppose you have found biomarkers, okay, for predicting something that is of interest to you. And then, can you use the same set of biomarkers in a slightly different context or to a different system, right? For example, if you have identified biomarkers based on, a mouse model, can it be applied to, predicting or making prognosis or diagnosis of a human disease, right? How does the gene regulations or pathways studied in one model organism be used to study, another organism? Can we transfer knowledge in a more rigorous and robust manner? Or how does a predictive model developed for one specific type of disease be applied to another type of disease or another class or another type of the same disease, right? So those are many of the things that motivate the study of transfer learning. Graphs and biological networks can be very useful for studying transfer learning in a more rigorous way. And I think we just started to scratch the surface of what we can do in the context of transfer learning using biological networks. And Arjun is going to give a couple of examples.
Let me actually skip this. So the two things that I want to highlight for transfer learning is this idea. Let me just introduce the first idea, which is pretty straightforward that all of us can think of intuitively, I think. If we know how to do network embedding, the way in which we can think about transferring, being able to learn from one network and predicting on the other, would be something like this, which is that you would run a traditional network embedding algorithm on, let's say, two different networks. Let's say we can take the example that Jun said. Let's say one network is genes interactions in humans and the other is gene interaction in some other species, let's say mouse. And the way we want to train models in one species and make predictions in the other would be to do network embedding independently on human network, independently on the mouse network, and then do something called alignment of the embedding space. So the way people do this is to say, in this network, can we identify something like a set of landmark nodes, a bunch of nodes that I can somehow assume that are exactly the same for all practical purposes. Maybe they perform the same function. This could be based on, let's say, one-to-one orthologs, right? And then what you can then do is to say, once I have learned a network embedding space for human genes and then one for mouse genes, you can then take this space and sort of do a linear transformation. And then so that you overlap these two spaces so that the landmarks align perfectly with each other. And that way you've essentially sort of put all the embeddings in the same shared space. And then you can learn in let's say a node classification task, you can learn new machine learning algorithms to classify human genes. Then once that model is trained, you can apply the same model to classify, let's say, mouse genes. Okay. The other idea, which is where this is an area that is super nascent, and there are very few methods that have actually been broadly applied. So this is really open for lots of applications. So here, this is at least theoretically, the idea in which we want to do everything simultaneously in some integrated way, where we want to embed jointly while maintaining reasonable alignment, right? But this is a pretty complicated task. And then where we want to be minimizing 4 different loss functions, right? The first, these 2 loss functions, LSA and LSB, are essentially our traditional network embedding loss functions. We want to make sure that the distance between the embedding vectors of very structurally similar nodes in the individual networks are pretty close to each other in the embedding space. This one basically says that I want to make sure that the corresponding, for the nodes that I know are corresponding across these networks, those embedding vectors are as close to each other as possible. And finally, if I want to do this entire thing in a supervised manner, I might also have some labels on, let's say, nodes only in graph 1 or only in graph 2. And I want to optimize the fact that these node embedding vectors become inputs to a machine learning algorithm. You train a new set of weights to make predictions on these node labels, and you want to minimize all these loss functions together. So this is a very general framework, but it's yet to be applied on a large scale to real networks. So this is pretty open. Okay. All right. This is going to be our final topic before our discussion. I think. Hi, Juan.
Thank you. And before I start, I just want to point it out that actually all the slides are collaborative work between Yang and I, and actually the whole group, and actually most of the slides are made by Yang. So I'm here giving the presentation, but all the credit goes to Yang. So I just want to, we've been talking about the whole day, networks and graphs. I want to mention that a lot of the biological molecules actually have a defined 3D spatial characteristics and requirements. For example, and it's important to know the 3D structure. For example, for small molecules, drugs, right? You have a lot of these stereoisomers, right? So if you look at their composition, they have the same exact composition. Pedro gave another example this morning, right? But in this case, there's really, if you just look at the composition of the different, they really have nothing, there's no difference. But because of this, how this 3D, spatial relationships between them, they are different, stereoisomers. One can be water soluble, and the other one can be poorly soluble, but can be an anti-cancer drug. Right? Another example of this is ibuprofen. We all take ibuprofen, but ibuprofen actually you take have 2 stereo isomers in the drug you take. Only the S form is active. The other one is just there for the ride. It's not active, right? So knowing this, 3D spatial relationship between all of the different, atoms and things like that is actually quite important. Same thing with protein structures, which is really what we do quite a bit. And Mona also mentioned about this, in her, she does a lot of this work as well. And everybody knows about this. Is 3D structures for proteins are very, very important, right? They form this, and we have now, AlphaFold, right? Can make a lot, The goal is actually to predict the 3D spatial structures of this. And of course, often the active sites, right, the highlighted, this pocket, the active site has a very defined shape, right? And I'm using the shape in a very general way. Not only this actually 3D shape, but also there is hydrophobicity, all the biophysical and chemical properties you can see, right, as the color indicates, right? So these 3D shapes and surfaces are quite important for protein function and how you can see, right, they complement the binding of this molecule, right? So all of these, and so there clearly when you talk about, did I miss a slide or something? Anyway, so when we talk about a very straightforward way is just use the regular Euclidean CNN, right? So you have this filter or this kernel go from 2D, right? So, and then you can go to 3D. But the problem is, and that's one of the topics I want to talk about, is the geometric. It's not, it doesn't, it's invariant to deformations of the 3D shape you're trying to actually learn, perform the convolution on, versus if you do, so this is using sort of Euclidean distance, Euclidean convolution, but if you want to do geometric, there is a time and place for that. But for geometric convolution, you can see, right, in this case, is the future shapes around the deformation or the structure of this particular subject. Okay, so this is sort of geometric CNN, geometric convolutions versus the traditional Euclidean-based convolutions. And so I think, again, we talk about graphs. Here I just want to very quickly talk about how do you generate from a, so if we have starting from a network is very sort of natural to go to a graph because a network is a graph. But if we just have a 3D structure, how do we go to a graph, right? And then there are two methods. One is so-called the input invariant, basically using the local coordinate, the relative local coordinates, which is, this is sort of what we do. And this is actually, I was introduced to this by Yang. This is so-called the model invariant or equivariant, basically using global coordinates, using a Cartesian XYZ coordinate. I will explain this using a recent paper. So you can create a graph from a 3D spatial relationships, and then you can do sort of a convolution, right, on this graph, create GNN or GCN, and then generate outputs. So for I would say traditional, the so-called input invariant graph, so from 3D structures, you can get all the atoms right in this structure, and you can figure out the distance, for example, between one atom to all the other atoms. You can put, we will talk about, you can put in even angles and all of that, and you create basically you create a graph, and you can put on for each node, each atom. You can also put on many other things like a convolution, like conservation, biochemical, physical chemical properties of the atom, and many other things, and you can do graph convolution. And as I said, you can consider distance. There are many different ways to construct, to go from a 3D structure to a graph, right? You can consider distance. You can consider distance plus angles and plus dihedral and all of this. And the things to consider is whether the representation, the conversion from the structure to the graph is complete, and how effective, right? All of these things to consider, and how effective is the representation, and whether when you do this convolution on this graph, the message passing efficiency and accuracy, all of these canonical problems. And the last thing I will talk about is this so-called model equivariant or invariant sort of way of generating the graphs. What that is that in the input invariant, however you rotate that structure, it doesn't change the graph, and it gives the same output. But in this way, basically, you can think about, for example, you're trying to predict a protein ligand binding, and you want to, basically, if the input rotates, your output also rotates correspondingly with it. So you basically can think about, you generate there's an XYZ Cartesian coordinates with this input, and you construct the graph that way, so that if you changes, the prediction also changes with it. There's, it's a paper on this, just published this year. And so that's, anyway, so I will stop there. And I think Arjun for other emergent problems.
I thought maybe we'll just pause and then take questions. And then if there is time, we can go to the other topics.
Okay, sure.
So we've been strongly encouraged to use the microphone when people are not in front of that one, so just an FYI. People online couldn't hear as well.
I will ask a question about the selection of neural network-based models, actually. So there is graph convolutional networks we use for simple models, but when the problem becomes more complex, we are using attention mechanism like graph attention networks, and then we can even go to message passing algorithms. So I just wonder, what should be the criteria, in your opinion, to select the proper GNN model? For example, there's a paper which suggests the second version of graph attention networks. In that paper, it is the how attentive are graph attention networks, something like that, the paper. And they are showing that with a simple modification, they are improving graph attention networks. Okay, for proving that, they are trying many data sets. But interestingly, for many data sets, they see that the models without attention mechanisms are better than those attention networks. So, in your opinion, what should be the criteria to select the proper GNN model? How to decide the complexity? Where to stop? I just wonder your thoughts about it.
Yes, there's no free lunch.
Arjun, you want to use the microphone?
No, I said that there's probably no free lunch. There is, I think without the context of what the problem is, it's pretty hard to figure out. I think, let me say a couple of things. I think one is the complexity of the model, at least the amount of training data or the way in which you are able to do unsupervised learning or the number of unlabeled data that you're able to use should also scale with model complexity in some way. Otherwise, it's probably not going to be possible to use more complex models. The other thing that I can say is from our experience, there are many cases where we have been trying to use things like graph convolutional neural networks and all stuff, and many times even these models are actually, they are baseline models that are based not on the original graph or not based on convolutional neural networks are actually sometimes very hard to beat if the problem is pretty straightforward to actually solve. So I don't have a better answer than this. I'm sorry.
Or there is not enough training.
Yeah,
Data
right.
set is very limited.
Right.
It's severely limited, right?
Yeah, that's a good question. I don't have an answer, but just want to make the question even more confusing. I think for, if you're thinking about GNNs, there are two levels of understanding. One is at the theory level, one is at the practical level. So at the theory level, you wonder how much, your networks are representative, can represent, right? The universal representation theorem says that if you have a deep learning neural network with enough neurons, it can approximate certain functions or its derivatives. So it's a representability. So that's a theory level thing. I think Peja showed this WL test for the graph isomorphism. So people designed the GIN, right, graph isomorphism network. So that's more addressing the theoretical representability issue. But the performance is not only by the representability. Representability is more like a theoretical upper bound. What you really reach is also dependent on the data. So there's another concept other than representability, that's a learnability. So a lot of the survey use different data sets, different assessments, and try different methods. It's not clear how much we can learn from them, which model is better. So that's just my 2 cents. Thank you.
Yeah, I think currently graph neural network is still at the very beginning stage. Different to traditional deep neural network. For traditional deep neural network, we already have very good understanding. Know the representation can give us very powerful results. But so far for graph neural network, it's still at a very beginning stage. For example, if we increase the number of layers, the result become poor, but not become better. So that's different to the traditional deep neural networks. So I think but this is still ongoing work, so in the whole community. So I think, this is a very good question. So I think there's a lot of work still need to be done to improve the graph neural network.
Can we please use the mic? Online folks were not able to hear us previously.
Yeah, I wanted to ask about the self-supervision approaches with respect to with graphs. I mean, obviously, self-supervision has been like a game changer in a lot of sequence realms, like in natural language, but also in proteins. And I was just curious with graphs, like, you know, especially with most biological networks, of course, there are many different types. The networks are just much smaller. And so the self-supervision part seems harder to do, at least in terms of molecular networks. So I was just curious what your thoughts were on that, or how that might work, or what domains in networks, biological networks, they would be applicable.
Thank you. Great question. I can try to start. You're right that self-supervised learning has made the tremendous progress in natural language processing. Lenore was talking about protein language models. That's one of the great applications. You talked about the length or the size issue. So I think the self-representation learning is more about learning the invariance in the data structure. So I'm not sure how much that's limited or restricted by the size. We recently worked on not small molecule graphs, but protein contact graphs. Now, we used to worry about the bottleneck of protein structures, but now with AlphaFold, we feel like we have a lot of structured data. So we have a lot of protein contact graphs nowadays. And one of the applications I studied recently was using the self-supervised learning to learn better the protein structure representation in graphs so that we can have a better downstream performance of predicting protein ligand affinity. So we do see some improvement, but there's still a lot of space to improve. Yeah, thank you.
So you mean like learning contact maps? Like what are good contact maps?
That is right. Yeah, there's something very interesting because you need to design the tasks very carefully. I still don't have the final answer for that. For instance, if you're just using amino acid as a node feature and try to predict what's the imputed node's amino acid, that task might be over supervision, and then you may learn some garbage knowledge, then that fails terribly in the downstream prediction. But if the self-supervision is too easy, that doesn't inject enough knowledge either. So there's something about what they call the curriculum of the self-supervision. That's still an open topic.
How do you perturb? So when you try to do this, the contact, I'm very interested in this. Right? So how do you perturb that? So you have these node structures, you construct your contact maps, right? And then how do you perturb that for your self-supervised learning?
Oh, so in the you think you're talking about a contrastive approach then?
Yeah.
In the predictive approach, it's about predicting what's not hidden from what's predicting what's hidden from not hidden. By perturbation, you are talking about the graph view or augmentation.
Yeah. So what's hidden, not hidden? I'm sorry, just maybe tell me again.
Oh yeah, okay.
What's hidden, what's not hidden? Yeah, can you explain that?
I see what you mean. Basically, okay, in both kind of methods, you could, hide, you can generate the different views of the graphs.
Okay.
For instance, removing some nodes or edges. We have a study on self-supervised learning, and we're looking at their performance on different graphs. Some are social, some are chemical graphs, some are protein graphs. And what we found was that the different self-supervised attacks or different augmentation or different views have different performance gain versus loss for different kind of for different kind of graphs. Which at the beginning it was just a lot
different
of
view. I'm sorry, just different view because for the image you show rotating, zoom in, zoom out at different. So for this contact graph.
right. You can drop a node, you can drop an edge, you can drop a small cluster or click. But then we found that, for instance, in some cases, it's like a edge dropping, which makes perfect sense for a social network. So edge dropping was found to help the social network predictions, right? But if you do edge dropping for chemical graphs, the performance was much worse. And we hypothesized that, well, that doesn't make any sense because when you're doing this perturbation, what you're learning is the semantics, right? You're learning what's invariant. So in a social network, if you drop just a few edges, that doesn't change the cluster identity and other kind of properties you want to predict. But speaking of small molecules, dropping an edge may mean a completely different, chemical bond or chemical that has different properties. So, so it's still early, but we have a lot of interesting observations and hoping to see more out of it. Yeah, great question.
Ben had a question.
Hi. So I was interested in the connection between the spatial geometric network section and the network embedding section in the following way. The network embedding is about starting with a network and then embedding it in some space that's convenient to then do further machine learning, et cetera. But then with the spatial geometric setting, you actually start in a convenient space. You start in the actual sort of physical space. And so I wonder, what's the advantage of then converting things into a network? Because then presumably, or maybe you would then go take that network and then re-embed it in some different space. And I wonder if you're going to do that anyway, why not just skip the network and go from one space to the other?
Well, the question is, how do you do that, right? So there are different ways. So the question is, yes, you could do, as I said, 3D CNN or geometric, geodesic convolution directly. That is one way of doing this. But I'm not so sure this is a space that is, easily can be learned, right? So the idea is, and we have found actually when we use, for what we want to do to predict protein binding sites, the GCN actually outperforms in our hands 3D CNN quite significantly for the implementation that we've done. So I don't know whether that. I'm not saying this is going to be better. I think you made a good point, right? So yeah.
Yeah, no, I mean, it's not about, better or worse. I don't know. That's not even my field. I'm just sort of thinking like kind of, what are the connections between these two? And I'm really, maybe is there a way to think about the representation problem that, you don't even need a graph, right? You're sort of in 3D space with properties and you just want to embed into some other convenient space.
So you could totally just straight using 3D treat. That you're right. So you could just treat the protein structure as a 3D image and just bam, go to the 3D CNN, and there you go. That's one way to do it. And the other way, of course, is to consider this as a construct a graph to capture the spatial relationships between atoms or amino acids, however you want to do it, and then to do the learning on the graph.
Do you think some of the advantage you're seeing is just because maybe the graph? Do you think some of the advantage you're seeing is just because the graph CNNs are, just better studied, and maybe the tool set works better, or the algorithms work better, or do you think there's some intrinsic thing about let's change the representation to a graph and then go analyze it?
I don't know. That's a good. Is there any other answer to that?
I'll take it.
Go ahead.
I think we're talking about a few different things here. There's kind of graph neural networks, graph convolutional networks like the version we have right now. Then there's 3D CNNs, which is like the 2D CNN but voxelized, which can also be used to analyze 3D objects. And then there's the things that we saw at the end, like the spatially equivariant models, and all those are doing really different things. I think 3D CNNs are really, really hard to work with, and they're critically dependent on like orientation and location in space. So you want to put in a protein structure, you have to stick it up the right way, the domain has to be in a certain corner, and if you flip that protein structure around, like there's no correspondence between the translated version and the original version. And that's been the critical advantage of this new class of the SE3 models. Maybe if you could flip ahead a couple slides. That equibind paper, like, it's not even working with just graph relationships anymore, saying here's a residue, it's connected to this residue because they're close together in the 3D space, but it's able to model distances and angles and things like this such that you can basically do reasoning on the representation of this 3D surface and flip it around and translate it and do all these things. So you can say things like, let's try to figure out where this small molecule sticks on the 3D surface of this protein, and I don't have to do all these tricks you would have to do with this 3D CNN, like try to augment your training data set by rotating it and translating it so that you attempt to learn those invariances and hope you get lucky during training. Instead, they're just baked in. So I think there is a difference between that class of models and what you get out of graph structures.
And there are some implementations of geodesic convolution is using polar axis on each on each point, so that to get this, so basically to using, yeah.
Okay, I have two, I think, simple questions. One question is, you talked about like the problems, basically linked prediction and node classification. What about module detection? And like, have graph neural networks been, or graph embedding, whatever methods been shown to be useful there? And my second question is about linked prediction. And the question is, for the test set, what can we assume? Does it, the test nodes, do they have to be connected to the training graph? Or can they be completely independent? And how do you deal with that? Like, do you encode it already beforehand? And what do you do with that?
I'll answer the second one first. Thanks. For link prediction, I think if I understand it correctly, if there's, let's say, a node, you're asking about a node that was not seen during training.
Link prediction, yes. Yeah, a node that is not training.
Right. Let me think.
If you say inductive, then you should be able to do it, right?
Right, exactly.
And when do you embed it? What do you do with that?
But I think that node needs to be connected to other nodes that were seen during training, right? Is that correct?
Let me try, and I think Marika will can correct me if I'm wrong. So if it's an inductive method, you can learn a transformation function which will use node attributes from the self node and the neighbors to learn the node embedding. Even that node doesn't exist in the training graph, it can learn, that function can learn the embedding. And the link prediction could be, you have the embedding of node 1 and node 2 in your test network, and then you can do dot product or some sort of similarity kernel function to compute the link between these two nodes. But I'll let Marika also say.
Yeah, so that's a great point, the great question, I would say. And so briefly, you don't need a test node to be connected to the training node. So the whole idea of graph neural networks was to try to make neural networks such that they can generate embeddings that are, in the simplest case, meaning GNNs, not equivariant models, invariant to the number of neighbors a node has. So, which is in sharp contrast to, say, CNN models or LSTM models used for images or sequences that always assume that an object, whether that's a word or a pixel in the image, has a fixed number of neighbors, which are pixels to the top, left, right, or word next to the left or to the right. So the implication of that is that GNN models can, for any node as input, if you give it a set of neighbors, and now you have a very flexible way how to define what the neighbors are. In the simplest case, neighbors are just direct neighbors. It can generate an embedding and can do that without consideration of whether those nodes that are direct neighbors were in your training set. Because for each of those direct neighbors, it can also create those embeddings for them, and it aggregates them to create an embedding for the node. So once you train this function f, which is a GNN, you can then use it in a new part of the network, or even a new network that has not been seen during training. Why? Because that will make an assumption. That makes an assumption that two nodes that have similar topology, and so topology now is very general function of what it means your local neighborhood is. Those two nodes will get similar embedding vectors, meaning vectors that are represented by points nearby in that d-dimensional embedding space that we learn. Then it depends on application what kind of similarity you want to capture. Depending on that, you would define different notion of what a neighborhood of a node is. And then this gives you different kinds of embeddings that one can then make different types of predictions for. The other parts of the question that you mentioned was related to community detection. And so I think it's quite interesting because there are very few applications of community detection in using GNNs, or the principle is such that we generate embeddings and then we cluster them with standard clustering algorithms. It's not as we see in other network community graph clustering algorithms that start with a graph and look for some densely connected sets of nodes. Instead, the idea here is I have a graph, I will learn embeddings for their nodes or subgraphs or edges or entire graphs. Once I get those vectors, it's very easy to do various linear algebra operations, so one can rely on standard clustering algorithms for that. So I'd say mostly the kind of tasks that are being solved in these techniques are node classification, link prediction, and comparing network elements, whatever you would like your network elements to be, nodes, edges, subgraphs, entire graphs. And these models particularly excel well at graph level classification tasks. Like, for example, what's mentioned here, where a data set is a large set of small graphs, or a large set of large graphs, or molecules, or proteins, which would then be graph level prediction tasks that these methods are quite very good at.
And if at all there is some notion of community detection can also be thought of as supervised node classification, where, for example, if there is some prior knowledge for some of your nodes for being part of certain communities, then you can make this as part of the node embedding problem. And then where you train your GNNs to also make sure that the tables are being classified correctly in addition to learning embedding, and then cluster.
So I have a question, and maybe also for Marinka, but going back to the link prediction. So in certain types of networks, as I mentioned, you have like higher order patterns with respect to annotations and topologies. Oh, sorry, did you hear the first part, or should I repeat? Sure, I'd be happy to repeat. So I want to talk about link prediction again, or ask about link prediction again. So when you look at like physical interaction networks, because you can think of all the molecular functions a protein does as kind of like different tools, like a screwdriver and a hammer or whatever, you kind of have certain patterns of molecular functions that work together in certain topologies. And so that's actually predictive of whether links are there or not. And so can you represent in these graph neural nets those kind of higher order annotations and topologies that would help you do link prediction?
Yes, briefly you can. Michael Bronstein from Imperial College London, now at Oxford, has a series of papers, including theorems on how higher order GNNs relate to random walks on hypergraphs, and how to design GNNs that do neural message passing on motifs, and how expressive they are relative to some of higher order kernels, like shortest path kernels or graph kernels or other type of graph kernels. So there is quite a substantial body of empirical work that can be done, as well as theoretical work that support this notion. Another comment that I'd like to make is that I feel in many applications that exist nowadays, we see, because the field is so young, we see direct application of methods that were not designed for biological networks to biological networks. In the context of physical protein interaction network, for example, several times today, there was this method mentioned known as L3, which essentially says if you have a physical protein interaction network, if you want to predict functions of proteins, you should not just look at the functions of direct neighbors, but the functions 2 hops away. And if they share the same function, and kind of the function of your nodes that are 2 hops away might inform the function of the target node, not necessarily the function of a node that is direct neighbor. So that is in sharp contrast with what generic GNNs would do, because generic GNN is based on the principle when I generate an embedding for node U, I will look at direct neighbors and learn some weighted function. In the simplest case, just average. If it's not an average, it's a weighted function attention of my neighbors when I learn an embedding for a node U. So that might work, for example, for social, for information networks, but for different kinds of biological networks, that's not really appropriate, that's not a necessarily appropriate mechanism. So what these models allow you to do is to specify your own message passing scheme, which essentially is a simple thing that has just 3 functions that one needs to specify, and then you have your own neural architecture that is designed for your task, which is for every node you need to decide what's my neighbor, who are my neighboring nodes. Then you need to decide, that's the first operation. The second one is, when I have 2 nodes that are neighbors, what do I share from one node to the other, which is a neural message. And then the third part is, when I have a node and I get all messages from my neighbors, then I need to aggregate them and update my own embedding. And I can do that through an average or through a weighted average and so on. So once you define these 3 functions, you have your own neural architecture. And defining those functions in a biologically meaningful way might really be interesting.
Does that require everything to break down into pairs? You have to be able to, so sometimes you have emergent structures that
Yes.
are ordered in pairs.
So if you have emergent structures, you can consider them using hyper GNNs. And there was another comment mentioned before about the depth of these graph neural networks, which I think it's a great point. It's a great comment, because what we see is that GNNs are rather shallow. What does that mean? We rarely see GNNs with more than 3 to 5 hidden layers. This is surprising in a way, because for other types of neural networks, we routinely see them having tens or hundreds of hidden layers. So why do we use shallow graph neural networks? So I think the problem here is, it can really be easily explained by the data that we see. Most biological networks are small word networks. So they have small diameters, typically less than 10, somewhere on the order of 7 to 10. So what's an implication of that? If you have a GNN with k hidden layers, that means that an embedding of a node will depend on nodes that are at most k hops away and nothing more. So that means effectively that if you set the number of hidden layers to the diameter of the graph, then every node in the graph could influence the embedding of any other node, right? So essentially this implies that it doesn't make sense to go much more than the diameter of the graph. But furthermore, from lots of kind of classic work, we know that most informative features for predicting node properties are 2 or 3 hops away. Like work from Anna Goldenberg in Toronto has shown that through many of the classic work in ML. So that effectively means that it's not useful to have a really, really deep neural network, because having features that are local or convolving information around local neighborhood of a node will already capture much of useful information. So there are lots of these kind of heuristics that can, or connections with graph theory that can help inform selection of hyperparameters.
My question was what questions or comments Marinka had, so great. Any other questions on this topic, which obviously we can have the whole workshop on machine learning and graphs.
So do you have to worry about the topology of the graph, as in scale-free networks versus regular graphs? And does that, if you have a hub node, do you need to maybe suppress it in some way?
Yes. So the default option should always be that you normalize them. When you send those messages along edges of the graph, you should always normalize them by the degree of the node. Without that, it's almost like what you end up having is just some kind of popularity vote. So that's just something that should always be there, because what it influences a GNN, because it tells it what it should focus on. And if you have a hub node with hundreds or thousands of neighbors, it will, because that node contributes so much towards the value of the loss function, a GNN will figure it out that if I want to decrease the value of a loss function, which is of course what I want to do, right? I should just optimize for the sole embedding of that hub node, which is exactly what we don't want to do. So we should kind of, yeah, I would suggest always normalizing by degrees.
So I was curious, as you've been surveying the literature, if you see anyone doing controls for these types of graph neural networks. Something obvious would be if you're going to claim that using a graph neural network instead of any other sort of baseline. I'm looking at Arjun, who talked about baselines. If the graph neural network is giving you a boost, then presumably it should be because there's real graph structure that's meaningful and being used by the graph neural network. And there's all sorts of control graphs you can generate to test that, just randomizing edges. We've done this briefly and we're surprised by what happened, but I'm curious as you survey the rest of the field what you're seeing.
Again, I can say a couple of things. So in some of the works that we've done, randomized graphs, especially if they are degree preserving, are actually, can give you relatively performance that's relatively close to the original graph. Especially, we have tried a bunch of different randomizations where we preserve no degree, or also where we preserve, we generate random graphs where we say the probability of a link in a particular random graph is simply the product of the probabilities, products that are of some number that's proportional to their degree. Right? And this is even harder to beat by random graphs. So essentially, higher and higher order node level properties that we are able to incorporate in the random graph, they simply give almost equal amount of information as the original graph, which is kind of surprising and a little bit sad. The other thing that we've also done is in the entire node embedding literature, especially when people want to take a network, do node embedding, and then do node classification, right? In this task, what we've done is to say, okay, instead of supplying the node embedding vector as the feature vector in some downstream supervised machine learning model, we can simply take the original graph and supply the vector that's like a row in the original adjacency matrix, so which is just the original graph. And actually, if you don't you have to tune the embedding hyperparameters a lot to get your downstream performance better than the original adjacency matrix. So this is also something that's not usually done in many node embedding or node classification papers also. So this is a baseline that we sort of try and implement a lot, but it's actually not common.
So I just want to follow up. This is a really good question. We didn't do a random graph as a control, although I doubt that will, because that basically just average, if it's a random graph, it's really for our protein interbinding site prediction, right? So you can see any of the protein functional site, you can clearly see why this kind of a neighborhood sort of have a message passing from your adjacent residues is useful, because any of the functional site is not one residue, right? It's multiple residues that have similar properties, like positively charged, that can accommodate a negatively charged residue in there from another protein. What we've done is we compare, so we have a previous methods that's not based, we use random forest, and then we use the exact same features, exact data set, but switch to our GCN architecture, and we can see the boost of the performance, right? So that shows that it's really from the GCN gave us. We can get more information out of the available features.
I think I'm arguing against that, actually. What if the GCN architecture with a shuffled graph also beats the random forest? That's the control. I did that test once on one data set, and it matched the real contact map graph for some protein-related problems. So now I'm curious about that whenever I read papers using GCNs elsewhere.
All right, we can try. We can try that.
I think that also goes back to, I think Ben was talking about this network only and then score only.
Yeah, that's part of what inspired my question.
Yeah. So for example, we have tried this problem of sample classification, where, transcriptome sample classification. So let's say you are given the expression level of every single gene in the sample, you have to predict something about the original sample. let's say, which tissue it came from, or the disease and all that. You can either use the expression vector as the input to the learning algorithm, or you can use the GCN to make a prediction about the sample, where you use the expression levels as scores in the individual nodes. It is, again, the original samples vectors are almost always better than using the GCN. So this is also something that we need to see, how to improve. So in my general opinion, the way we are at least using graphs right now, especially graph neural networks, in some way is to not essentially to just boost performance, but to see if we can at least go in the direction of similar performance, but somehow more interpretability, because we are inducing some kind of biological context by providing this graph to this one.
So one thing I'd like to add to what Arjun just mentioned is, I also feel that the use of graph neural network topology in modeling and prediction is not necessarily the boost of the predictive performance. However, the reproducibility and the robustness of the predictions we actually make based on the model. So I think it is very, based on my experience, it's always very useful to think about, okay, if I take this graphical topology into this predictive test, and if I don't necessarily aim to boost the performance, but then maximize the reproducibility on independent data sets, what is the result? And my answer has been very positive. So for example, it's very easy to, beat the performance of GCN based on, very simple or traditional models if we focus only on the predictive performance. However, if we test the reproducibility of the prediction results on independent data sets, that also goes away. So meaning, if we use these graph structures, what we tells us something about the underlying biology, and use that to regularize the data set, a very noisy data set that we have, and which is very shallow compared to the complexity of the system we're dealing with, you probably can gain much more in terms of the robustness or reliability of the predictions. And of course, that is tied to the interpretability of the features that we generate. So I thought that that's something that we should bear in mind.
Last question, Teja.
So just a quick question. I think maybe we have something more to talk about, generally evaluation of network-based prediction methods, because there's so many traps that Lenore was mentioning, and we can talk about it. But I have one about the graph neural networks and embeddings. And we start with relational data, and then we embed every element in that data into a point, and now treat it as IID data. It strikes me like something is still missing in there, and I might be wrong. What happens? Why do we treat it data that way? Is this the right way to do? Yeah, that's my question.
I'm not sure I understand.
So you start with the data that is relational.
Yes.
Okay, so let's say the nodes are patients, and maybe one is a family member of another, right?
Yeah.
These patients can also have properties.
Right.
Now, you embed these nodes suddenly, and you somehow have now eliminated these family relationships that was actually important. And now we treat these individuals not as relational data, but as IID data, standard machine learning, and as Marinka was saying, we are clustering them after embedding. I feel like something is missing there, or so I don't know. Just kind of asking for clarifications.
So, in one of our current studies, we do very similar to what you explained. So we get the embeddings of patients from a patient similar to network, and then after that we do subtype prediction. And when we just use embeddings, it works, but if we add the features of those individuals, again, like a kind of a residual things, if we add those raw features that we used in the network, again, then our performance increases actually. So adding those integrations at the later stage of the downstream task really helps. So the node embedding uses those node attributes, learns something, but doesn't learn everything. At least in our example, we observe that.
In the interest of time, let's thank everybody, and not just the current team, which thank you so much, but all 4 teams for today.